An intellectually enriching day one, filled with insightful keynote lectures, technical sessions, and vibrant academic discussions, we now move forward with renewed enthusiasm into the day two of prestigious scientific gathering. Yesterday's deliberations laid a strong foundation by exploring sustainable approaches in drug discovery, advancements in nanoparticulate drug design, delivery systems, protein-based therapeutics, and the vital role of technology transfer in bridging research with real-world application.
Day 2 promises further deepen our understanding through advanced scientific sessions, expert interactions, and meaningful academic engagement. The continuation of interdisciplinary dialogue reflects to the evolution of landscape and pharmaceutical sciences, where the sustainability, innovation, and translation research to shape the future of healthcare.
We are deeply honored by the continued presence of our esteemed dignitaries and speakers whose expertise and guidance inspire us to pursue excellence in research and innovation. Your valuable insights is not only enrich this symposium, but also motivate researchers and students to contribute meaningfully to the scientific development. To all the participants, delegates, your active involvement and intellectual curiosity truly define the success of this international platform. Let's make the most of today's sessions by engaging in thoughtful discussions, fostering collaborations, and strengthening professional networks. Once again, I warmly welcome you all to the Day 2 of International Symposium. May today's proceedings be equally enlightening, productive, and inspiring. Without further ado, let's move into the scientific sessions. It is now my privilege to introduce our esteemed co-chairperson for this session, Mrs. M. Rekha Ma'am, Associate Professor, Department of Pharmaceutics, Hindu College of Pharmacy. Ma'am has over 17 years of rich experience in teaching and research and has published more than 20 review and research articles in reputed national and international journals. She has also published 4 patents and authored books in the field of industrial pharmacy. She is a life member of IPA and APTI, and her research interests focus on solid lipid nanoparticles and the advanced drug delivery system. We are honored to have her as a co-chair for this session, and we warmly welcome you, ma'am. Now we welcome Mrs. Rekha ma'am to introduce our chairperson for this today's session.
Good morning, everyone. So myself, Sri Rekha, Assistant Professor in Department of Pharmaceutics in Hindu College of Pharmacy. It gives me an immense pleasure to introduce today's session chairperson, Dr. A. Kishore Babu Garu. Sir is a principal professor at Vagdevi College of Pharmacy. And sir completed his PhD from JNTU University, Hyderabad. And sir has, to his credit, sir published 10 patents and grants in Indian, German, and South Africa. And sir is also published 34 research and review articles in various national and international peer-reviewed indexed journals. And sir published 1 textbook. And sir is also an author for 2 elsewhere chapters. And sir guided 15 PG students and 43 UG students, and sir is guided 2 PhD scholars, and sir organized various SERB grant funded projects, and sir is a reviewer for various journals, that is Curious Journal of Medical Journal Science, International Journal of Research and Analytical Reviews, World Journal of Current Medical and Pharmaceutical Research, and sir is also a editorial board member for JCPR, JIAPS and JCSCR, and sir, to his credit for his publications, have the citations of 331 with a H index of 12, and sir is a member of APTI and IPGA and APP, and sir is also a well well expert in administration and curriculum designing. With this brief introduction, I welcome you, sir. Thank you for the opportunity.
Yeah, thank you, madam. Very good morning, all of you. Actually, it's my immense pleasure to read about a keynote speaker profile. He is Dr. Jitendra Patel. It's my pleasure to read your profile. Yeah, Dr. Jitendra Patel is currently serving as Associate Professor at Datta Mega College of Pharmacy, Maharashtra. He completed his PhD in Pharmaceutical Sciences from the Jawaharlal Nehru Technological University, JNTU Hyderabad in 2017. And after earning his M.Pharmacy in Pharmacognosy and B.Pharm with a distinction, actually he is having more than 15 years of experience, academic experience and research experience too. He has served in the reputed various institutions, including JNTUH affiliated colleges, Avanti Institute of Pharmaceutical Sciences and Geetham School of Pharmacy. And his research focuses on pharmacognosy, phytochemistry, and natural products, as well as the nanotechnology-based drug delivery systems and translational pharmaceutical sciences. Dr. Patel has published more than 50 research articles in indexed journals and reputed Scopus and Web of Science and Springer, Bentham journals. He has authored books also and book chapters with the international publications, including Elsevier and Lambert academic publications. At the same time, he is having the H index of the 10 Google Scholar Index over the 350 citations. He has received the research and conference grants from DST, SERB, New Delhi and CSIR as well as Government of India. And also he was awarded the Young Scientist International Travel Support by SERB, DST. He is also a recipient of the State Talent Award. As a dedicated academic mentor, he has guided numerous B.Pharm, M.Pharm, PharmD and PhD scholars and actively contributes to curriculum development, NAC, NB accreditations process and research mentoring. Dr. Patel continues to contribute significantly to bridging a natural product research with the modern nanomedicine, aiming to translate phytopharmaceutical innovations into a sustainable healthcare solutions. It's a very great profile, Dr. Jitendra Patel. A warm welcome to you. And I would like to thank to the management, secretary and correspondent of Nirmala College of Pharmacy, as well as the organizing secretary and the principal. He's my friend, Dr. Pamal Reddy Garu. Thank you very much, all of you.
Good morning, all. Yes. Play video. First, I will. This is the university where I am working right now. So you just have a glance of. Datta Megi Institute of Higher Education and Research. It is situated in Wardha, Maharashtra, Nagpur, right? So in that institute, we are having dental, medical, physiotherapy, Ayurvedic, nursing, right? So in that context, we are having so many collaborative projects in one campus. In the now that university is going to be Adani University because Adani has taken over that. So I think there is a technical problem. Okay, fine. You just open the PPT. No issue. We'll play afterwards.
Onward march of this university. DMI-HER is a deemed university offering over 270 multidisciplinary programs to more than 10,000 students. Away from the hustle and bustle of the city, the campus is a self-sustaining complex. It also has an off-campus college at Nagpur. The infrastructure-rich, full-fledged medical colleges, along with large hospitals, facilitate hands-on
Hospital,
learning.
1500 bedded hospital.
Allied sciences students have an array of non-medical streams to choose from. The
Cancer
students
hospital
benefit
separate.
from leading national and international collaborations and the opportunity to partner with some of the world's best institutions. Exchange programs and combined research initiatives empower the students to grow and gain greater recognition. With a proven track record of graduates and postgraduates in 25 plus countries, DMI-HER students are prepared for a global career. The environment is gender equal and rich with cross-cultural interactions with students from numerous countries and continents coming together. The campus offers a
Yoga
complete
hall, it is
life
there in
to
the
its students with facilities for
Swimming
sports, workouts,
This all
simulation
within the
labs,
campus.
research house, literary society,
Separate research
New
house.
Age auditorium, NSS activities, and more.
Dental.
With 661 plus full-time faculty, 795 plus PhD scholars, and 48 plus fellowship students, DMI-HER offers one of India's most leading and evolving research.
Okay, PPT. Right. So today my talk is based on the nano formulations, right? Nanotechnology. Like, how many of you know about the nano? What do you mean by nano? Yes, participants, students. What do you mean by nano? Small, right? So when we reduce the size, right? when we reduce the size of the particular active entity which is going to play a role to give the therapeutic efficacy, that becomes nano. So nowadays, the scenario is going on in nanotechnology. Of course, बीमारियां बढ़ रही हैं, बीमारियों का साइज बड़ा हो रहा है एंड दवाइयों का साइज छोटा हो रहा है, right? So in that context, nano career mediated how we are going to have the tannin and flavonoid. So you might be studying like tannin and flavonoid. These are the two important phytoconstituents which play an important role, right, in therapeutic efficacy. So here I am going to cover the hepatoprotective, right, how it is going to have the liver protection, right? So in that context, as we know, the global burden, it is increasing day by day for the river-related ailments or the disorder, right? And there is a huge gap, research gap, we can say, that is there between the existing medicine and the traditional medicine. So somewhere in the, maybe in the bioavailability, somewhere maybe in the poor solubility. So there is a need of the formulation to have the better solubility, better bioavailability. That's what there's a need of to go for the nano formulations of those the phytoconstituents so that we can have the better efficacy, right? So my the purpose of talk is to highlight the potential and right. So in liver, whenever the metabolism, because so many cases like chronic disease, chronic liver disease, अगर कई बार क्या होता है कि पेशेंट को लॉन्ग टाइम के लिए मेडिकेशन लेना पड़ता है, राइट? द पर्सन हैज टू मे बी इन द केस ऑफ डायबिटीज, मे बी इन द केस ऑफ हाइपरटेंशन, मे बी इन द केस ऑफ ट्यूबरकुलोसिस. So when we are in the long-term medication, every day we need to consume some medication, and here and there that will impact onto your liver because of the first pass metabolism, second pass metabolism, right? So that metabolism can cause the damage of hepatocytes. So that may be one of the reason, right? Biological may be virus, bacteria by the different route, what we eat, what we consume, what we inhale, right? There are so many routes where the microbes can enter into your body and it can cause liver damage, right? And the physical cause like trauma, radiation, right? So these are the different causes where metabolic cause like genetic disorder or right, this kind of fatty liver disease, right? So these are the damage where it will be in the cellular level or it may be into the molecular level, right? So in cellular level, it can go for apoptosis, necrosis. These are the process where it damage the liver, right? This is the pathophysiology of liver damage, how whatever the things I told like fibrosis, right? The reactive oxygen species which will cause the oxidative stress. So these are the different mechanisms where the liver is getting damage, which the person will never know. Sometimes what happens is liver, whenever it will damage, it will not show the sign and symptom. Whenever liver will damage up to 60%, 70%, then also the patient will not be able to know that his or her liver is started damaging. The hepatocytes are getting collapse, right? So by the time the person will know that liver has started damaging, it may be too late. That's what whenever we go for the body checkup, definitely we need to check the liver function test. That is very important. SGOT, SGPT, total bilirubin, right? It is not only the cholesterol. Nowadays people are only bothered about the sugar and BP, right? I don't have sugar and I don't have BP. That means I am fine. Nothing is going to happen to me. So that is also the part of your full body checkup. Liver is very important organ and this has to need this kind of follow-up, right? So as we know that in our pharmacognosy also, there are so many crude drug you guys are studying, biological source, chemical constituents and uses, right? Herbal drug technology is there in B.Pharm. You are studying or not? Biological source, chemical constituents and uses. Yes, students, I am talking to you. Pharmacognosy. How many of you study pharmacognosy? Raise your hands. Pharmacognosy and phytochemistry. Pharmacognosy 1, pharmacognosy 2, herbal drug technology. You are studying now? Yes or no? Once again, yes or no? Okay. What you study in that? Chemical constituents, biological source, uses. In that uses, so many times you are studying like carminative, purgative, laxative, hepatoprotective, anti-inflammatory, right? In that context, these are the medicinal plants you might have heard, turmeric, ginger, these are already ginseng, these are already there in your syllabus, right? And you will be surprised that these are the drug which has already there in the research, and the researchers, the scientists, they are getting the phytoconstituents from those crude drugs which is there in your syllabus, and they are formulating different types of nano formulation to protect the liver and so many other disease, right? So when we talk here, I am talking about the nanotechnology. So there are so many formulations which we can formulate with the phytoconstituents to have a better absorption, first of all, right? Until and unless the phytoconstituents, the phytochemical constituents which you study, if it will be into the nano size, it can have the better absorption, better distribution, and a better efficacy. That is the concept of nanotechnology, right? So we may have different nano emulsion. We can go for liposome, we can go for dendrimers. There are so many other formulation available, right? So which will help into the protection of the liver by the different mechanism like oxidant defense, anti-inflammatory, cellular repair mechanism, right? So liver may damage in many way, multiple ways. So in those way, if your phytoconstituents is helping in the any of the mechanism which is causing the damage of hepatocytes, that means it is working, right? So this is the mechanism of hepatic injury, like the formation of oxygen or reactive oxygen species, maybe disturbance into the cell signaling. At cellular level, it may have the necrosis, it may have the apoptosis, right? So like that, the cells are getting damaged, like your hepatocytes, your liver cell, these are getting damaged like that. Right, so this is the mechanism of oxidative stress, how it is going to happen. It may have the internal triggers and external triggers, right? Up the question is, liver कैसे खराब होता है? मेरा liver खराब हो गया. My liver is damaged. Whenever, now it is happening in the small kids also. You don't think that it may happen to the old age, 40 plus, 50 plus, 60 plus, grandfather, grandmother, only they are suffering from liver related issues. It is not like that. Nowadays we know that what is our food habit. All the junk foods are your favorite. Noodles, pizza, burger, biryani. How many of your favorite biryani? Raise your hands. All. Right? So you know that liver is the organ which will decide the which food should go inside, which food should not go inside. Trademarks will be done there. That is the first stoppage where it will be decided. So maybe to satisfy your taste buds, you may be eating all the unhealthy, let me say, things, right? So slowly your liver will start damaging and you will never know. That's what it is told, like you should have all the healthy fruits and vegetables, right? Why it is told? Because it may have the flavonoid, it may have the plant phenols, it may have the so many other nutrients which can protect your liver in daily basis, antioxidant properties. That's very important. Why we are here, the mechanism, it is having the antioxidant, right? So these are the different reasons where these kinds of reactive oxygen species are getting formed. what is the solution nowadays? Because every time the person who is suffering from liver disease, maybe hepatitis, cirrhosis, jaundice, or the non-steroidal, right? So what is happening? So what the solution, right? Whenever we go for the it may have the liposome. Suppose we are going to formulate the liposome, what it is, how it is going to address? It is going to address. It is going to enhance the bioavailability absorption, right? Ultimately, when the drug is going to have the better bioavailability, we can expect the better, what do you call, action, better effect of that particular phytoconstituent which we want to deliver at the proper site where it can show its action, right? Then solid lipid nanoparticle, it may improve the stability of the particular drug. Nano emulsion we can have, which is going to again address the solubility. Time release, when we are planning for control release of that phytoconstituents, we can go for the polymeric nanoparticles, right? So these are some case study where the garlic and all other curcumin, which are the popular phytoconstituents, which has been studied for its better efficacy, right? Why it is required to have the what do you call, together, right? There is a terms called a synergistic effect, right? When the one drug can enhance the efficacy of the another drug, right? So this synergistic effect that we need to address, like these kinds of study has been already done, like in case of cancer, in case of diabetes, when they come together, they are showing the therapeutic effect, right? So this is the mechanic pathway of the, now as the topic of my talk is to address the tannin and flavonoid, right? So you might have heard a different tannin related drug in your pharmacognosy, yes or no? Tannin, myrobalan fruit, Arjuna, yes or no? Arjunolic acid, arjunolene, you are studying or not in pharmacognosy, right? Then different flavonoids we are having, plant phenol. In herbal drug technology also we are formulating phytosomes, liposomes. It is there in the B.Pharm syllabus. It's not the rocket science which I am dealing here, right? B.Pharm syllabus, it is there already. You are studying in herbal drug technology, formulation of phytosomes, right? So like that, it may have the different tannin. For example, here I am focusing the tannin. So tannin fraction, if because so many research has been carried out in last few decades, like tannin are responsible to protect the hepatocytes, right? So if you see the 10 year ago, the paper used to come in alcoholic extract, or maybe in the what do you call, the kind of crude extract, we used to follow the activity, biological activity, like alcoholic extract having the hepatoprotective activity. But nowadays, it is not the extract, we need to fractionate it. Fractionation, there is a word, right? You might be doing the column chromatography, right? It is there in the syllabus, again, phytochemical investigation. So in column chromatography, what we do, we separate the phytoconstituent, right? Alkaloid, glycoside, tannin, flavonoid. When we will be able to separate from the extract, the efficacy will increase. Yes or no? The efficacy will increase. So likewise, we are going to have this kind of particular which is responsible, like ellagic acid. Suppose in tannin, ellagic acid is responsible for hepatoprotective activity. And when we, with the tannin fraction, if we prepare the what do you call, nano formulation, it may have the better efficacy. You can see the gallic acid, ellagic acid, and the proanthocyanidine. So these three constituents address different types of problem, hepatotoxicity problem, right? Different constituents may address different types of hepatotoxicity. Like it may give the gallic acid antioxidant activity due to the free radical scavenging. Anti-inflammatory, again, and then anti-apoptotic, right, which is there, inhibit the oxidative stress and support the liver detoxification. So all these constituents are having different role. You can see here, if the gallic acid, it will be for formulated in liposomes. It can target the passive targeting in the particular hepatocytes. If ellagic acid, if it will be formulated in the polymeric nanoparticles, it can have the active targeting like a ligand preceptor interaction, which is very useful, which may not be possible with the crude extract, which may not be possible with the other formulations, maybe in the tablet capsule or so many other formulations, right? So that's what it is the need, right? So these are the main flavonoids. The another parts apart from tannin, the flavonoid is also reported to have the hepatoprotective activity for last 1 decade of the research paper. If you see that all the flavonoids, they are targeting to have the formulation for hepatoprotective, right? So what are the main flavonoids like which may have, right? So quercetin, as we know, the quercetin is the very good example of flavonoid, which is very famous for the hepatoprotective activity. Apart from silymarin is one of the standard also, which we are using for the hepatoprotective activity of the different plant extract. We are using silymarin as a standard, yes or no? So camphorol, like, so these are the luteolin and epigenin and nigrinine, right? So these are the main major hepatoprotective flavonoid which are being isolated and screened for last few years. When it will be formulated into the particular formulation, right? Say for example here, it is the liposomal encapsulation. So in liposomal encapsulation, it may have the hydrophobic bilayer and encapsulated flavonoid. So you can see inside this, there is a chemical structure. That is the flavonoid which I told you in earlier slide. So this flavonoid will be encapsulated and in bilayer, the flavonoid will be there. So whenever it will be targeted to liver, we can have you can see the hydrophilic and hydrophobic. So based on the requirement, the availability of these phytoconstituents into the liver side, it will enhance, and at the same time we can have the proper bioavailability. Here also you can see the liposomal flavonoid uptake in hepatic cell, how it is going to have the first endocytosis, how it is going to reach to the address to the mitochondrial protection, fuse the membrane, and it is going to enter to the hepatocyte. Ultimately, it need to address the hepatocytes, the liver cell, because the damage is there in the liver cell. So it need to address the liver cell, right? So these kinds of advantages we are going to get, right? And why the necessity of the nano formulation and the traditional crude extract, that we need to know, right? So there is a two comparative pharmacokinetic free versus liposomal flavonoid, right? So if you are using free flavonoid, right? If it is free flavonoid, it may have the so many disadvantage, right? It is not going to absorb properly, first of all, right? As I told you, hydrophilic and hydrophobic bilayer, the coating of the phytoconstituents, right, which is ultimately is going to pass the metabolism, right, the rapid metabolism in liver, you can see here, right, if it is free. If you are encapsulated in liposomal flavonoid, it may enhance the gut uptake immediately, first of all, and poor absorption in gut if it is free, and when it will be you can see here slower liver metabolism and the reduced renal clearance. Here it will be here fast renal clearance, so that is also disadvantage. Your phytoconstituents, the main chemical entity which is responsible for hepatoprotective activity, that need to address the problem. It is not just going to travel in your body. It need to address the problem up to significant level. It has to rectify the liver cell damage, right? So that's what the lower systemic circulation, and here you can see the sustained release into systemic circulation, how it is going to have for long term sustained release, right? Here it may have the lower systemic circulation. So that is the advantage when we formulate this into the nano formulation. Apart from this, we are having the genomic instruments and the technologies in research for this kind of formulation. Like you can see at the genetic level how it is going to have to to address the therapy or personalized medicine where one individual body is different, one individual body responding different, right? It's not that the particular liposome has been given the very good result. The same age group and same gender, it may give the same result. It may not possible. So in that case also we are having this advanced research also in gene expression and all those. It's not that the nano formulation will be always giving the very good, excellent effect. It may have some disadvantage also, right? As the recent research, it has explored like exposure route, it may be right, exposure route and toxicity evaluation steps. So this we need to address, even toxicity evaluation for the nano material which we are going to use, in vitro toxicity. So toxicity study, it is here more compared to your, anyway we are doing the toxicity even for the crude extract also, in preclinical stage also, or in the clinical stage also, because we know in the clinical trial in phase 1, phase 2, we are more concerned about the safety rather than the effectiveness, right? So that's what it become very important for the risk assessment to know, because so many drugs, so many nano formulation may give the side effect after launching into the market, after consuming by the patient for the long term, it may give the serious side effect, right? So that's what the risk assessment is very important over here, right? So how we can conclude this kind of formulations? There is a need, like we know that tannin and flavonoids are proven to have the hepatoprotective for the recent years. And if we will be able to fractionate them, if we will be able to separate them from their main extract, and when we are going for the nano formulations, I think we will be able to address better to the hepatotoxicity, right? Because we know the delivery limitation, ultimately the goal is to have the solubility, bioavailability, and their clinical efficacy. If the drug is soluble, better soluble, and if it is reaching to the systemic circulation, the bioavailability is better, definitely it will be able to address the hepatotoxicity at a better level, right? Nano carrier solution is the one of the nano formulation, enhanced solubility. It is enhancing the targeting and the therapeutic performance. That's what it is important to have the nano formulation, right? Improve the modulation of reactive oxygen species and other enzymes, inflammatory pathway. Safety consideration require careful evaluation. Sustainable nano phytomedicine integrating holds a strong translation potential. If you have any question, please raise. If anybody is having any questions, I will be able to answer.
Any query, questions? Yeah, thank you. I think so. You don't have any questions, right?
Thank you, Dr. Jitendra. I have one question. Just tell our students regarding the nano formulation and its regulatory issues.
Yeah. Regulatory issues, yeah, it definitely it may have the main burden, the main hurdle, we can say, whenever any drug or any formulation, when it comes to the market, it is not the samosa or biryani, right? where we can sell into the market without having any license, without having any approval, without having any authorization, right? Whenever any drug, it gives better efficacy, good effect, it will be having another side also. It may give the good side effect also. It may give the good toxicity also. Yes or no? So the regulatory body will be always looking for the formulations to have better safety rather than the effectiveness. Yes, of course, effectiveness is very important. That is what the research and development is going on, to have to address the problem in effective way and significant way. But regulatory hurdles, always it will be looking for the long-term side effect. If the drug, if the nano formulation is giving the side action into the long term, definitely it required a proper pharmacovigilance, so that it can have the traceability, and the drug can be stopped manufacturing, and the life-threatening event we can stop over there, right? So that's what, of course, nano formulation and nanotechnology is having boom, and it is going to have the better efficacy in the different disease and disorder. But at the same time, regulatory need to also be very strict, and it need to monitor at micro level. Yes. Thank you.
Yeah, thank you, sir. Thank you very much. Actually, it is.
So, regarding phytosomes, so we are interested to carry out the phytosomes formulation and also.
Okay.
So there is a herb-drug interactions. Some combination means sometimes we used to face the stability problems also, herb interactions, all like that.
Yes.
And that is the one. How can we overcome that kind of? And another question is phytosomes, if we form framed means, formulated means, then what are the basic tests for that? To confirm the phytosomes formed, and the stability studies parameters, where we can go for that confirmation of the phytosomes are stable, like that availability of the labs in and around.
Yeah.
The possibilities are there, sir. Thank you for this.
Thank you for the question. Actually, it is very good question. Phytosomes, like, actually, one thing I will clear, madam, before going to your question. Phytosomes is actually thread. Actually, it's liposome. But nowadays, so many research paper it is coming by directly coating the phytosome. Since we use the phytoconstituents, we say it is phytosomes. Generally, actually it is the lipid bilayer to address the poor solubility and to address the bioavailability, of course, right? So your question was on to the stability, right? So whenever phytosome form, there are certain parameters which we need to, just like other nanoformulation, like short-term study, long-term stability testing, right? How we do for our other formulations, maybe in the solid dosage form. So here also we need to see, like, by keeping into the temperature, by keeping into the proper pH, and by keeping into the humidity, relative humidity, right? Maybe for the 1 month, 3 months, 6 months, that will go on, right? But in hurry, when we do the preparation of phytosome in our laboratory, what we do is we immediately rush to the animal study, right? Like, suppose you have prepared for anti-diabetic. So first we want to know whether it is giving anti-diabetic or not. And if it is proving anti-diabetic, immediately we will rush for the publication. Our hurry will be in that way, even for the any research scholar or whatever they will be, right? And once it is published, and then later we came to know, like, after 1 month it is not able to be stable, right? It is having some temperature related issue, it is having the, like, it is cracking, or it is changing the color, it is changing the, it's a stable form, right? So that's what it is important to address that in the different zone, like temperature zone, and we need to check it by the certain, like, drug release, that maybe the cracking, the how it is going to release the drug, and all those things need to be checked, and that required a certain laboratory help also in case of suppose if it is not available in one particular institution. So in that case it is required. Thank you.
Thank you, sir. Actually, your talk is very nice. Whatever you selected, the topic is very good nowadays. Everybody are thinking about major organs other than the liver, how the heart is working, how the cardiac problems, and so everybody are focused on that. Nobody are bothered about the liver nowadays. So whatever you are selected, the topic is good. The liver damage, liver disorders, especially coming to the what are the main reasons behind the liver diseases, and herbal formulations, especially drug delivery systems, novel drug delivery systems. You took the liposomes. Yeah, it is the compatibility and very good drug delivery system for that. And the role of the tannins and flavonoids, how they will be acting as a greater role, which is fight against the liver disorders. So you explained very nice with all mechanisms. Thank you. And I think so this a good informative session which helps to the students. Thank you very much to giving this opportunity.
Thank you.
Thank you so much, sir, for such a thought-provoking session. We are so enlightened with your session. First and foremost, we express our deepest gratitude to our eminent resource person, Dr. Jitendra Patil, sir, for an enlightening and thought-provoking section.
As a token of gratitude, we now request the dignitaries to kindly felicitate our honorable resource person, Dr. Jitendra Patel, sir.
We request the HODs to kindly join the dignitaries for the felicitation of our resource person. We also request the Srinivas Reddy Bonam sir and Nagbhushanam sir to kindly join the dignitaries for the felicitation of resource person. A sincere thanks to our chairperson, Dr. A. Kishore Babu sir. Your presence added depth and stature to the session, and we are truly grateful.
May I request our chairperson, Dr. A. Kishore Babu sir, to kindly accept the token of gratitude from our dignitaries.
We are all especially thankful to our co-chairperson, Mrs. M. Srirekha ma'am, for presiding over the session and for offering valuable insights.
As a token of gratitude, we request the dignitaries to felicitate our co-chairperson, M. Rekha ma'am.
Once again, we are really thankful for the resource person, chairperson and co-chairperson for a great session. And now we are having a high tea break for 10 minutes. After the high tea, we will be having the session by Dr. Srinivas Reddy Bonam sir, Senior Scientist, Vaccine Immunology Laboratory, Department of Applied Biology, CSIR. So I request all the students to go for the high tea break for 5 minutes, and after within 5 minutes the session will be started. So again all of you gather in the seminar hall.
ಜಾರಿಂದದಿ ನಕ್ಕ ನಾ ಒಳ್ಳಂತ ಆಡಿಂದೆ ಕೈತಕ್ಕ
I request all the students to please gather in the auditorium. The next session is about to begin. The next session is very informative session, so I request all the students to please gather in the auditorium.
Facilities for sports, workouts, simulation labs, research house, literary society, new age auditorium, NSS activities, and more. DMI HER offers one of India's most leading and evolving research ecosystems. Highly advanced facilities, grant opportunities, seed fund support, co-working space, advanced computing facility.
I request all the students should be gathered in the auditorium.
I request
That was truly an energetic performance. Give again a big round of applause for the listed performers. Next, we have Ashwita with a solo performance from second year B Pharmacy. Ashwita with a solo performance from second B Pharmacy. Let's invite her with a
I request all the students must gather in the auditorium.
that was truly an
The session
energetic
going to be started.
A reminder for the participants of cultural programs, submit your team names to the hosting department and also your tracks to the technical department. Students who have completed their high tea, gather in the auditorium. The session is going to be started. I request the discipline committee members to send all the students into the seminar hall. The session is going to be started. Once again, I request the discipline committee members to send the students into the seminar hall. The session is going to be started. Welcome back to the second session of day 2 of today's International Symposium on Sustainable Drug Design and Innovative Nanoparticulate Development, Protein and Technology Transfer, sponsored by CSIR India, organized by Nirmala College of Pharmacy. I now take the honor of inviting our esteemed co-chairperson for the session, Dr. N. Venkat Deepak Sir, Assistant Professor, Department of Pharmacy Practice, Chebrolu Harmaya Institute of Pharmaceutical Sciences, to kindly come forward and join the session panel.
We are deeply honored to have you with us and look forward to your valuable guidance during the session.
I request our co-chairperson to introduce the session's chairperson.
Good morning, all of you. It's my immense pleasure to introduce Dr. M.V. Nagabhushanam Garu, Chairperson. Dr. M.V. Nagabhushanam Garu holds a PhD degree from Acharya Nagarjuna University and an MBA from CSM Institute of Graduate Sciences, Canada, and B.Pharm and M.Pharm degrees from Birla Institute of Technologies and Sciences, Pilani, and 18 years of academic and 7 years of research experience. He worked at Natco Pharma Limited and ICI Pharmaceuticals before serving as associate professor at Viswabharati College of Pharmaceutical Sciences, and currently sir is professor and principal at Hindu College of Pharmacy. Sir has published 20 national and 134 international papers and presented numerous national and international conferences, guided 25 B.Pharm and 20 M.Pharm and 4 PhD scholars, and is a recognized research guide at JNTU Kakinada, Hyderabad, and Acharya Nagarjuna University and Krishna University. With this brief introduction, I welcome Nagabhushanam sir.
Good morning to all dignitaries. It's my pleasure and honor to stand before you to say and also to introduce Sri Srinivas Reddy Garu. He is an immunopharmacologist, graduated from Chalapati Institute of Pharmaceutical Sciences, Acharya Nagarjuna University, Guntur, and sir has graduated B.Pharm in 2011 and M.Pharmacy in 2013, and received a PhD in 2018 from Acharya Nagarjuna University from Academy of Scientific and Innovative Research at Indian Institute of Chemical Technology, IICT, India. His sir's PhD is focused on vaccine adjustment development against infectious diseases. During his PhD, sir was awarded Raman Charpak Fellowship by CEF IPRA, Indo-French Center for the Promotion of Advanced Research, for 6 months to work under the guidance of Professor Muller at the Institute of Biologic Molecular University of Strasbourg, France in 2017, where he worked on therapeutic strategies for autoimmune diseases, including P140, acting against systemic lupus erythematosus. From 2018 to early 2020, Sir worked as a postdoctoral researcher with Professor Sylvain Muller, later continued postdoctoral research on various aspects of immunology, including regulatory T cells and basophils, the next generation flu vaccines for the world, Indigo, and COVID-19 with Professor Jagadish Barry at INSERM. And he then moved to the University of Texas in the Department of Microbiology and Immunology, where Sir was trained under Dr. Hu, where he developed novel mRNA vaccines for emerging infectious diseases, including COVID-19 and therapeutics. At present, Sir is a senior scientist at CSIR Indian Institute of Chemical Technology, Hyderabad, and also holds honorary assistant professor position at ACSIR, Ghaziabad, Uttar Pradesh. Sir's current research focuses on development of mRNA-based vaccines and therapeutics, and has published more than 90 research and review papers and 7 book chapters, and has 5 patents to his credit. Sir has licensed 1 pharmacology and pharmacy and editorial board member for PLOS1, Frontiers in Immunology, Journal of Immunology Research, Molecular Biology Reports, Frontiers in Molecular Biosciences, Frontiers in Drug Delivery, and Journal of Young Pharmacists. Sir has been serving as a guest editor for journals Vaccines, Frontiers in Immunology, Journal of Immunopharmacology Research, and others. Sir's publications have received more than 4,000 citations, and with a 35 H index and 60 I-10 index. With these few introductory words about Sri Srinivas Reddy Garu, I thank Dr. Pamal Reddy Garu, Principal, and Sister Nirmala Jyoti Madam, Secretary and Correspondent of Nirmala College of Pharmacy, for giving this opportunity. Thank you all, and I request Srinivas Reddy Garu to start the session.
Thank you, sir. First of all, thanks Dr. Roy for this invitation, and thanks to Professor Bamal Reddy sir for organizing such a wonderful event. Yeah, this is my third time to visit to this college. So, why don't you students come forward? 15 minutes, I don't take much of your time. All backbenchers come forward. We'll go fast. Yeah, all backbenches, occupy these first chairs. Come, please. All backbenches. This side, left side. Boys, come on. So you people are not interested, so you wanted to stay there only, right? Okay. So again, I wanted to thank Jitendra Patel because Dr. Jitendra Patel gave a very vast introduction on what are different forms of nanomaterials, liposomes, solid lipid nanoparticles, emulsions, everything. So that made my life very easy because I'll be presenting two aspects here. One is an oil in water emulsion. The second nano form is mRNA LNP. So I will be continuing his talk actually. So this is my academic and research journey. And if you see where I started, from pharmacology, and where I am now at present, CSR IICT. And I have been a team player in all these products. Now, during my PhD, we have licensed one vaccine adjuvant to Bharat Biotech, that is called Uthroside B. This is a natural saponin. And during my first postdoc, I worked on a molecule called P140, which is now in clinic for autoimmune disease, for treating autoimmune disease. And the second postdoc, I worked on flu vaccine development. Now the vaccine is in phase 2 clinical trials. And during my third postdoc, I worked on mRNA LNPs and as well as HIV, anti-HIV drugs. So with this expertise, I joined CSR IICT. So this is a very old slide and but useful slide. We have a lot of different vaccines in the market. They are in different forms. During the COVID, what we experienced, mRNA LNP vaccines are inactivated vaccines. So I'll be discussing mostly on mRNA LNP vaccines. And any vaccine, when we are taking, we need an adjuvant. Adjuvant is a substance which boosts the innate immune responses. So I worked in different aspects of these vaccine delivery systems to enhance the innate immune signal and adapt to immune responses. That I worked with Dr. Sampath Kumar during my PhD. So as I mentioned, I'll be discussing on oil in water emulsion. So this is a global consortium under Horizon 2020 funding. So where our global consortium target was making an effective and affordable flu vaccine for the world. So across Europe and across USA and India, many institutions are involved in developing these vaccines, and IICT also part of the IICT is also a part of the consortium. what we did, in the consortium, we have a company called Lightvax. They have developed an oil in water emulsion. This is the composition. Oil in water, you know, it's a quite familiar term, right, in pharmaceutics. So this is the composition, squalane and between 80 and PBS as well as CMS. This is the active ingredient in this emulsion. So then I was asked to dissect the mechanism of action of this oil in emulsion, how it is delivering the vaccine antigen and how it is effective in human cells. So what we identified, this emulsion is non-reactogenic and non-hemolytic, and it is compatible with biological system. And also this vaccine has shown effectiveness against influenza as well as malaria. Then when I took over this project, I wanted to know what is the chemical structure of this CMS and what is the composition of this emulsion and what to do next. So what I did quickly, I collected blood from the healthy donor, from blood bank. Then in the blood I have purified PBMCs. From PBMCs I have cultured monocytes. Monocytes differentiated to dendritic cells. Through these dendritic cells I have treated a vaccine, because my intention is to check how this emulsion is working on innate immune cells. So after treatment I realized that whether this is activating the innate immune system or not. Then I identified the molecules like CD80, CD86. These are surface receptors of these dendritic cells. So any activation of these dendritic cells, if the emulsion or if the formulation is inducing the innate immune signal, then these are the markers to identify, 80, 86. So we could see that the emulsion is inducing the innate immune signal. There is a significant enhancement. Then the next question we asked was whether this is because of squalane, because oil is there, or this is because of CMS, active ingredient. Then we used only CMS, the chemical active ingredient, and then with different doses we have treated the same dendritic cells. And we could see there is an increase and dose response we can see here. Again the same markers we used, 80, 86, and these are the predictive markers for looking at dendritic cells activation and maturation. Later what we did, so this emulsion is working, but what happens in terms of when we add antigen? So we have used the same PBMCs and purified the PBMCs, and we have used our H1N1 vaccine. This is called antigen, purified antigen, and then along with or without our emulsion. Then continued for culturing for 6 days. After 6 days again we looked at how this emulsion with or without antigen boosting the immune responses. So we could clearly see that here antigen specific immune responses with emulsion, and with there is no response without emulsion. So here also we could clearly see this bar represents the increased surface markers on dendritic cells. So later again dendritic cells are activated, fine, but how they are discussing, how they are communicating to T cells, because as I showed in the first figure, innate immune signal, then T cells. So the interaction should be there, otherwise we don't get enough immune responses. Then what I did, after activating dendritic cells, I have co-cultured the same autologous T cells to the activated dendritic cells to see the interaction of DCs and T cells. So when we co-culture these DCs and T cells, we could immediately dissect the different T cell polarization, Th1, Th2. These are different T cells present in our body, but that we are mimicking in the plate. Whether treatment of our vaccine can boost these type of immune responses. So again with our emulsion with antigen vaccine, we could clearly see this bar graph. So there is a significant increase in upregulation of T cell phenotyping markers. So after that study, then company immediately understood the mechanism. Okay, this is activating dendritic cells and T cell polarization. Then they finished the phase 1 clinical trials on this emulsion. Particularly, they focused on the older adults, safety and immunogenicity. Now it is in phase 2 clinical trials. So overall, due to interest of time, I am not showing a lot of data, and you don't understand also if I present. So the summary is, we used oil in water emulsion with H7 flu antigen, recombinant antigen, and then this emulsion is phagocytosis by the dendritic cells, and when these dendritic cells activate and mature, then they present the signals to T cells, then T cells polarize. So this is a summary. Similarly, we, as I told you, one saponin we worked on, I worked during my PhD. This is extracted from this Solanum nigrum, and this has shown very good immunopotentiation against dengue antigen. So after showing this data, immediately Bharat Biotech wanted to take for the their products. So we have transferred the technology to Bharat Biotech. So, and why we wanted to develop this, there is already a marketed product by GSK called QS21. But synthesis of this molecule is too tough. And the extraction, if you want this molecule, you need to cut down this big, huge tree. And it's like it's like a burden. So what we immediately looked for the alternative. So growing these small plants is easy rather than cutting bigger ones. So that is an alternative. And the second, good point is the limitations. Though this is effective against vaccines, but it is having hemolysis as well as mechanism of action is unknown. So what we did, we extracted this and we proved the mechanism, and we have shown that this is less hemolytic than the GSK product. So that's the reason BBL showed interest. So the second aspect is mRNA LNP. So this is the emulsion part. I completed. The second one is mRNA LNP. So when I joined my third postdoc at UTMB with Professor Hu, he has been working with Professor Drew Weissman, who is a pioneer in mRNA LNP vaccines. So then I joined his lab here. Then I learned the technology. When I joined, after the post-COVID, I joined his lab. So during that, my joining time, he was working on development of COVID-19 vaccines. So we know everyone, like spike is the most targeted protein for mRNA vaccines. But here in India, we took all inactivated vaccines. So similarly, I started understanding what is mRNA, because I know how immunology, what is immunology and how to test vaccine, but I don't know how to develop a vaccine, particularly mRNA vaccine. Then I started understanding what is mRNA. Okay, we know that DNA to mRNA and mRNA to protein. So, and how it is, mRNA is translating to protein and how it is going to degrade. Then I started understanding. Then later, I also started understanding how to protect this. mRNA is so sensitive. DNA is stable and protein is stable, but mRNA is not stable. So how people have been using technologies to protect this mRNA. So this is the technology where they are using called lipid nanoparticles. As Jitendra told already, liposomes. This is a form of liposomes called lipid nanoparticles. What we do here, we use all the lipid components, including ionizable lipid, helper lipids, cholesterol, and PEG lipids, and we use our mRNA, and these two we use a microfluidic device to formulate. This is called nanoassembler, microfluidic device. Through this device we make LNPs. Now the mRNA is protected, it is inside and it is protected from the RNases. And we do the dialysis followed by, if you treat this mRNA LNP to cells, so we could clearly see how these LNPs are going through phagocytosis process, and what happens after the release of mRNA, and how the translation happens, how the targeted protein expression happens. So this is the mechanism of mRNA LNP and the targeted protein expression. So knowing this concept, then we wanted to develop a novel mRNA vaccine. So we know that two are the competitors. One is SM102 Moderna, and the second one is ALC0315 Pfizer. So we wanted to see, we are also working on mRNA LNP. There are already products. So then we need to come up with a novelty, because those days, again, we know that a lot of variants are emerging in terms of SARS-2. So then our vaccines are not working, what developed by Pfizer as well as Moderna. So we were looking the literature. So what is the other target that we can choose? So we came up with a paper that published in Nature. What they identified, people who infected SARS coronavirus 1 20 years back. So they have shown a T-cell memory in their blood. So then we got an immediate idea. So why don't we use this N? N is the nucleocapsid protein. So they could able to detect the memory response from these 20 years back infected patients. So why don't we take this as a target and develop the vaccine? So then we immediately cloned that mRNA N into our vector, and then we purified the plasmid, and then we linearized. From linearized DNA only we will get mRNA. So then we prepared mRNA, in vitro transcribed mRNA, and then purified the mRNA, and then encapsulated into LNP to protect the mRNA degradation, and then evaluated for the expression of this mRNA, and in vivo also. So when we have our mRNA LNPs ready, so we immunized the mice, same at 0th day, like priming, and then followed by booster after 3 weeks. Then, because we all took 2 doses, right? Similarly, we just used the same strategy. So priming dose and booster dose. After 2 weeks of booster, we harvested the spleen and blood. Blood is for humoral immune responses, to look at the IgG, how it is antigen specific IgG. And spleen is for immune cell, to look at the immune cell interactions. We can see here our strategy N has worked out, and we could see antigen specific TNF alpha and IFN gamma, as well as N specific IgG. And if this is a mock, only LNPs, and this is with mRNA N, nucleic acid mRNA. So this is fine, but when we see neutralization titer, means our intention is vaccine should neutralize the virus. So we don't see here, because this N is inside the virus, it's not a surface protein. Then the next strategy what we used, and also we have seen the protection efficacy with mRNA N in hamsters. What we did here, we primed as well as we gave a booster dose to hamsters. After 2 weeks, we directly challenged with the virus. Then we could see whether hamsters are protected against this virus. So if you see the viral load after 2 DPA and 4 DPA, mRNA N immunized hamsters are having less viral load compared to mock, because mock is not protected, no vaccine. So mRNA N is having less vaccine. But still this is above the threshold. So then we wanted to go with the alternative strategy. What we did, we have now considered S also, because spike is already available in the market. So S plus N, how this strategy is going to work. So in this strategy, we could clearly see 5 out of 5 hamsters definitely knock down the complete virus. So there is no virus at all. So then we discovered that S plus N combination is a best strategy for coronavirus treatment. And the entire study goes like this. So we have identified the novel target from SARS-2 protein, nucleic acid, and we cloned into mRNA vector. Then we encapsulated mRNA into lipid nanoparticle, and we injected into the mice. Then when we inject it, nanoparticles goes through a phagocytosis process, where it presents protein in 2 directions. One is MHC class 2 pathway, and the second one is MHC1 pathway, where cytotoxic T cells directly kill the infected T cells, and where the exported protein presented to dendritic cells, processed by dendritic cells and presented to T cells, followed by humoral helping to the B cells. So this is a overall summary of this vaccine. And we can also see, again, I'm not showing entire data because later we also discovered how after injecting LNPs immediately in the mice in 1 day, less than 1 day, what are the changes happening in immune population that we looked at here. So I'm not showing the data, but the overall mechanism is like this. When we inject mRNA to the mice, in the less than 24 hours, we could see the activation of innate immune cells. That's the reason we all get fever after vaccination. Many of kids, doctors prescribe paracetamol to the kids. Why they prescribe paracetamol? Because this is what happening. When we inject any vaccine in 24 hours, the innate immune cells activate and release IL-1 beta, and IL-1 beta goes to hypothalamus and disturb the thermoregulatory sensor. So, but this is essential phenomena. If this innate immune activation is not there, then it won't help for the humoral immunity. And our booster dose of 3 weeks of booster dose, this enhances the antibody production. So this is the overall summary. And again, we have published all these works in good scientific, decent journals. Then when I joined at IICT, so I used the same strategy because what we learned there in abroad, so I started working, so I brought the technology. So as I worked with him, so the same nanotechnology I'm using now, but we bought the machine, which is around 1.2 crores. Now we have in my lab this equipment, microfluidics equipment, to make this mRNA protected LNPs. But we are working on different aspects because we are working on novel lipids, how to make these lipids, because they are mostly abroad, patent-owned lipids. So we wanted to find out indigenous lipids, novel lipids from Indian side. So we are working on novel lipid development as well as different vaccines we are working, because the template is same, LNP technology is same. If you change the protein, you get the novel. Either if you put cytokine, if you put vaccine, or if you put any protein replacement, the technology is same. So that's the reason we have started working with different aspects here: monkeypox, Nipah, and influenza, as well as antimicrobial resistance pathogens like Pseudomonas aeruginosa, and also some cancer vaccines we are working. And recently we ventured into CAR T cell therapy because this is a unique treatment for cancer. And these are the portfolio of products, and we are here almost. And our lab has been funded by Ignite Life Science Foundation, ANRF, as well as Bharat Biotech, and ICMR, as well as CSIR. So that's it. This is my last slide, and these are all my mentors, and this is my team now at CSR IICT. 4 PhD students and 2 project scientists and 3 project assistants. Yeah. Any questions? I'm happy to answer.
Yeah, very nice talk, sir. Of course, your research right from the abroad to India, and you are working in the vaccine or immunization sector. One of my doubt or even question also,
please.
There is a talk, and even in social media or in news also, like those who have taken the vaccine during COVID-19, maybe first dose, second dose, third dose, now the heart attack is coming in young age, and most of the heart attack, it is quoted like if you have taken the corona vaccine, it is correlating by the different platform. So what do you feel in this context? Is it really connected to that, or it is something else? Yeah.
Yeah, good question, Jitendra. So what we have been discussing with my collaborators even in abroad, people in abroad, they are not facing this issue. People in India, we are facing this issue. I don't know the reason, but nobody has dared to work on this aspect, because then we have to pull out, if it is a vaccine, you have to pull out the company.
Yeah.
So that's why it is more common in India. Even I witnessed this during, even in nearby my institutions. People who are active and who are sports, players, they are actually, we can see their sudden death and because of this. And they say that he is healthy and nothing illness, but still sudden death.
Sudden death and
People, they speculate this, but we don't have evidence to prove that. So somebody should invest some money and prove that this is because of this.
okay.
Unless
But it is
otherwise-
not proven yet.
Not proven yet. That's why we cannot give any statement on this.
But out of all these contexts, what is your personal feeling or opinion? Because you are working in same domain.
As you said in your talk, always two aspects:
Yeah.
efficacy aspects, toxicity aspect. There is no drug without side effect.
Yes.
This is because of vaccine side effect, or this is because of their genomic their gene level. We need to go with their gene profile. Genetics and combination of this vaccine, we don't know. These we need to dig out.
Maybe or may not.
Yes, may not be. Because abroad, I didn't hear this news. Abroad, when during the initial days of COVID-19 pandemic, as when they were using AstraZeneca vaccines, they know that the vaccine is inducing coagulopathy, means blood coagulation.
Yeah.
That's, they ruled out that is because of vaccine.
Okay.
But that also, few patients, they clearly told, even company also declared, yes, this is because of vaccine.
So vaccine this much long-term ADR maybe happen because we also taken so many vaccine during our childhood.
Yes.
Or right. So
Yes.
we never know like.
True. It should not have, because it's a foreign material. It's not our body protein. Always vaccine means we are taking protein from the virus or a pathogen. So our immune system will tackle it as a foreign protein, foreign material, and try to dismantle it. It should not affect our health.
Right.
So we need to go through why it's happening in Indian population at least.
Right.
Yeah.
Thank you.
Thank you.
Thanks, Dr. Srini, for your explanation.
Yeah.
I have one small doubt.
Yeah, please.
There are several cases we saw non-specific antigens in our body.
Yes.
Non-specific.
Yes.
So how the T cells can be activated so that they can catch any kind of antigen in our body?
A good question, but this takes again. Oh, close. Can I have my slides, please? I'll show you so that people will understand. Yeah, this slide. If you see, Dr. Roy, this is the mechanism what happens in our body in case of any foreign material if you put it in our body. If it is a vaccine, if it is a protein, if it is a virus, whenever enters the virus, because if you see, generally we have flu, every seasonal flu, but we don't take medicine. Flu also the same virus, but it tackles, immune system tackles the same way, and it processes this virus into the protein and present to the T cells. But your question is, what happens if it is an autoantigen? These T cells are actually trained in thymus. That's why we call this a T cell. During the thymus evolution, if the T cell is missing the proper processing, then that T cell emerges what we said tolerant deficient T cells, because all our T cells have tolerance to our immune system, so they don't detect. That is a tolerance mechanism existing in our body. But they skip this tolerance mechanism during the evolution in the thymus. That T cell only will recognize those are people more prone to autoimmune diseases.
Suppose if T cells are not recognizable, we
Yes.
are not able to catch the antigen.
Yes.
Then how we can externally we can ask them to catch the antigens?
You have to engineer the T cell. That's what we are working on CAR T cell therapy, because their cancer is not recognized, because cancer and generally it's not a pathogen, it's our own body's system. But protein is mutated or modulated, but it's a self protein. So now what we are doing, we are raising an antibody against that protein, whatever mutated protein, and we are engineering the T cells to recognize this mutated protein. That technology is called CAR T cells.
Antibodies.
Yes, antibodies. But antibody, whole antibody you cannot give, because again that have a adverse effect because it is having Y-shaped shape, and the Fc fragment is toxic. So we use only SCFE portion. Those FE portions we transduce to the T cells, and we engineer the T cells to express this SCFE portion so that it can recognize only particular protein of interest with a small epitope. It won't affect other cells. But this is costly affair. Each patient has to pay 40 lakh in India. Now it's there in the hospitals. If you ask Manipal, Apollo and everywhere, cancer patients who are affordable, if they pay 40 lakh, they are having treatment for any cancer.
So in this area, there is a lot more work need to do.
Yes, definitely. Yeah.
Thank you.
Thank you.
Sir, I had a small doubt.
Yes, ma'am.
So like all the vaccines,
Yes,
the patient will get resistant to this vaccine, what we will do? It's like
Patient, you see, this is entering in our body only 2 times. As I told, we are giving priming dose, booster dose. If you remember, my child or your child, less than 6 months age, we have the different schedule to give the vaccines. But later in life, we never give the vaccines, right? So that is the power of immune system. They recognize their once in life, and they keep the memory cells in our bone marrow.
Okay.
So whenever next time they encounter the similar type of pathogen, for example, the best example is polio vaccine. We took only one time. We never take in our entire lifetime later. But those memory cells are stored in our bone marrow. Whenever next time if we face this virus, our bone marrow immediately push the cells to the bloodstream, and they can recognize and kill the virus. That's always happening in our body. For flu, for seasonal flu also, we don't take any medicine, but immune system can tackle that.
Okay, sir.
Thank
you very much.
Thank you.
On behalf of all the delegates, faculty members, it is my pleasure to congratulate and thank Srinivas Reddy Garu for giving a wonderful session on the nano vaccines, from mechanistic adjuvant science to mRNA LNP therapeutics, from Vaccine Immunology Lab, CSIR, IICT. All our delegates as well as the participants are fortunate enough to listen to you, sir, having an international and exposure in world-renowned laboratories, having an excellent doctoral and postdoctoral experience in this vaccine research. Sir has explained about the vaccine adjuvants and effective and affordable flu vaccines for the world, as well as antigen presentation and antigen-specific immunoresponses. And it is an excellent work in the sense it is a collaborative work with the Bharat Biotech and ICMR, CSIR, collaboratively with world-renowned scientists. So all the delegates and the participants definitely would have enriched with the session, and which is evident with the question answer session. There is a lot of questions which were raised actively from the participants in this question answer session, which shows the excellency, the presentation and clarification of the doubts from the participants. We thank from Nirmala College of Pharmacy, Srinivas Reddy Garu, for being with all of us for his wonderful presentation. Thank you, sir.
Thank you so much, sir, for such a wonderful and thought-provoking session. First and foremost, we express our deepest gratitude to our eminent resource person, Dr. Srinivas Reddy Bonam sir, for an enlightening and thought-provoking session.
As a token of gratitude, we now request the dignitaries to kindly felicitate our honorable resource person, Dr. Srinivas Reddy sir.
I request the HODs to kindly join the dignitaries for the felicitation of our resource person. I request all the students to give a great round of applause for Srinivas Reddy Bonam sir for such an enlightening session. Thank you so much, sir. A sincere thanks to our chairperson, Dr. M.V. Nagabhushanam, sir. Your presence added depth and stature to the session, and we are truly grateful, sir.
May I request our chairperson, Dr. Nagabhushanam sir, to kindly accept a token of gratitude from our dignitaries.
Thank you so much, sir. We are especially thankful to our co-chairperson, Dr. N.V. Venkat Deepak, sir, for presiding over the session and offering valuable insights.
As a token of gratitude, we request the dignitaries to felicitate our co-chairperson, Dr. N. Venkat Deepak sir.
Once again, thank you so much, sir, for such a great session, and thank you, dignitaries. Now we all pause for the lunch, and please proceed to the lunch area behind the conference hall, and please kindly seat it back in the hall by sharp 1:25 p.m. for the next session. We kindly request all to kindly seat it back in the hall by sharp 1:20 p.m. for the next session by Dr. Raghavendra Ashoka Bahura sir, Senior Principal Scientist, Grand Med Limited, Galway, Ireland. And a reminder for the participants of cultural programs, give your names to the hosting department and submit your tracks to the technical department.
Ah.